Good morning. This is part 1 of module 3, the human nervous system. The human nervous system is the most complex system that controls various functions of the body and coordinate them. The brain and the spinal cord comprises the main division of the nervous system, that is the central nervous system. The peripheral nervous system consists of the neurons and parts of neurons found outside the central nervous system, which includes sensory neurons and motor neurons. We have discussed already this in the first module. Action potential of brain. The interconnections between neurons are called synapses. All synapses occur at or near cell bodies. They do not touch each other but do come into close proximity so that the axon of one nerve can activate the dendrite or cell body of another by producing a chemical, mostly acetylcholine, that stimulates the membrane of a dendrite or cell body. In some cases, the chemical is produced by one axon near another axon to inhibit the second axon from activating a neuron with which it can normally communicate. Because of the chemical method of transmission across a synapse from axon to dendrite or cell body, the communication can take place in one direction only. There are really two kinds of communication across a synapse: excitatory and inhibitory. The same chemical appears to be used in both. In general, several axons from different neurons are in communication with the input of any given neuron. Some act to excite the membrane of the receiver, while others tend to prevent it from being excited. Whether the neuron fires or not depends on the net effect of all the axons interacting with it. Electrochemical action of neuron. Neurons communicate with each other via electrical events called action potentials and chemical neurotransmitters. When an action potential reaches the presynaptic terminal, it causes neurotransmitter to be released from the neuron into the synaptic cleft, a 20 to 40 nanometer gap between the presynaptic axon terminal and the postsynaptic dendrite. After traveling across a synaptic cleft, the neurotransmitter will attach to neurotransmitter receptors on the postsynaptic side, and depending on the neurotransmitter released, which is dependent on the type of neuron releasing it, particular positive or negative ions will travel through channels that span the membrane. This is shown here in the figures. The effects of the various neurons acting on a receiving neuron are reflected in changes in the graded potentials of the receiving neuron. Graded potentials are variations around the average value of the resting potential. When this graded potential reaches a certain threshold, the neuron fires and an action potential develops. Regardless of the graded potential before firing, the action potentials of a given neuron are always the same and always travel at the same rate. An excitatory graded potential is called an excitatory postsynaptic potential, EPSP, and an inhibitory graded potential is called an inhibitory postsynaptic potential, IPSP. Neuronal communication. In neurons, these action potentials are of very short duration and are often called neuronal spikes or spike discharges. Information is usually transmitted in the form of spike discharge patterns. These patterns, which are simply the sequences of spikes that are transmitted down a particular neuronal pathway, are shown in the figures. The brain generates rhythmical potentials which originate in the individual neurons of the brain. These potentials get summated as millions of cells discharge synchronously and appear as a surface waveform. The EEG signal can be picked up with electrodes either from the scalp or directly from the cerebral cortex. The peak-to-peak amplitude of the waves that can be picked up from the scalp is normally 100 millivolt or less, while that on the exposed brain is about 1 millivolt. The frequency varies greatly with different behavioral states. The normal EEG frequency content ranges from 0.5 to 50 Hz. The variations in EEG signals, both in terms of amplitude and frequency, are of diagnostic values. The frequency information is particularly significant since the basic frequency of the EEG range is classified into the following 5 bands for purpose of EEG analysis. Delta 0.5 to 4 Hz, theta 4 to 8 Hz, alpha 8 to 13 Hz, beta 13 to 22 Hz, and gamma 22 to 30 Hz. The figure shows the 5 bands of EEG signals. Types of electrodes. Electrodes transforms ionic currents from the cerebral tissue into electrical currents. For a more localized measurement, the action potentials of a single neuron can be observed either extracellularly with a microelectrode located just outside the cell membrane or intracellularly with a microelectrode actually penetrating the cell. We will discuss the common electrodes which are used for measuring the EEG. Scalp electrodes. These are small metal discs usually made of stainless steel, tin, gold or silver covered with a silver chloride coating. They are placed on the scalp in specific positions. These positions are specified using the International 1020 system that we will be discussing later. Each electrode site is labeled with a letter and a number. The letter refers to the area of brain underlying the electrode. For example, F for frontal lobe, T for temporal lobe, etc. Even numbers denote the right side of the head and odd numbers denote the left side of the head. The diameter will be usually 4 to 10 millimeters. Some cup disc electrodes have hole in the center through which the electrolyte can be introduced after it is attached to the scalp. Another method uses a gel that can hold both electrode in place and provide good electrical contact. Many recording systems use a cap into which the electrodes are embedded. This facilitates recordings when high density arrays of electrodes are needed or when comparing recording sites. The second type is needle electrode. They are sharp wires made of steel or platinum. They are inserted into the capillary bed between the skin and the skull bone. It provides better signal quality than scalp electrodes, but they are having disadvantages that the patient will suffer from pain or infections. The next type is sphenoidal electrode. They are basically used to detect the epileptic activity in the temporal lobe. The patients are sedated for the procedure and the electrodes are placed with the aid of a needle into the region of the cheeks. The needle is removed and a thin wire is left in place and taped to the skin. The electrodes are easily removed upon completion of the evaluation. These electrodes can record electrical activity from deep parts of the temporal and frontal lobes. The patient feels some discomfort during the insertion and for several hours afterward, particularly when yawning or chewing. The risk of using sphenoidal electrodes are rare and almost always minor. A small amount of bleeding may occur during the needle insertion, but it is rarely a problem. Other risks include the possibility of infection or of a tiny piece of the bare wire remaining in the cheek. Nasopharyngeal electrodes are plastic tubes with a wire inside that ends with a blunt metal tip. The tip is around 2 to 5 millimeters diameter, uninsulated metal bore. The electrodes are inserted into the nose until the metal tip reaches the upper back part of the nose. Tip is placed against the roof of the nasopharynx and close to the tip of the temporal lobe. It may cause discomfort and slight injury to the mucous membrane. Subdural and epidural electrodes, shown in the figure, consist of sheets or single column strips of evenly spaced disc electrodes embedded in a thin layer of flexible, translucent elastic rubber. Sheets are inserted through craniotomy opening. It can also be used to stimulate the underlying cortex by applying an electrical current to individual electrodes. Depth electrodes are those electrodes which are placed within the substance of the brain. It usually consists of a bundle of fine wires that terminate at different cylindrical contacts along the length of the depth electrode, thus allowing recordings from the different depths. Usually platinum metal is used for MRI compatibility. Surgery is needed to place these electrodes. Placement of electrodes for EEG measurements. Since clinical EEG measurements are obtained from electrodes placed on the surface of the scalp, these waveforms represent a very gross type of summation of potentials that originate from an extremely large number of neurons in the vicinity of the electrodes. Originally, it was thought that the EEG potentials represent a summation of the action potentials of the neurons in the brain. Later theories indicate that the electrical patterns obtained from the scalp are actually the result of the graded potentials on the dendrites of neurons in the cerebral cortex and other parts of the brain, as they are influenced by the firing of other neurons that impinge on these dendrites. The 10/20 EEG electrode placement system. Placement of electrodes on the scalp is commonly dictated by the requirements of the measurement to be made. In clinical practice, a standard pattern called the 10/20 electrode placement system is generally used. This system, devised by a committee of the International Federation of Societies for Electroencephalography, is so named because electrode spacing is based on intervals of 10 and 20% of the distance between specified points on the scalp. Each site has a letter to identify the lobe and a number to identify the hemisphere location. There are 21 electrode locations in the 10/20 system. The 10/20 EEG electrode configuration is illustrated in the figure. Electrodes are identified according to their position on the head. For example, FP for frontal polar, F for frontal, C for central, P for parietal, and T for temporal, O for occipital. Odd numbers refer to electrodes on the left side of the head, and even numbers represent those on the right, while Z denotes midline electrodes. There are two types of EEG recording modes, monopolar and bipolar. In monopolar technique, EEG may be recorded by picking up the voltage difference between an active electrode on the scalp with respect to the reference electrode on the earlobe or any other part of the body. In bipolar technique, the voltage difference between two scalp electrodes is recorded. The basic block diagram of an EEG machine with both analog and digital components is shown in the figure. A pattern of electrodes on the head and the channels they are connected to is called a montage. Montages are always symmetrical. The reference electrode is generally placed on a non-active site such as the forehead or earlobe. EEG signals are transmitted from the electrodes to the head box, which is labeled according to the 1020 system, and then to the montage selector. The montage selector on analog EEG machine is a large panel containing switches that allow the user to select which electrode pair will have signals subtracted from each other to create an array of channels of output called a montage. Each channel is created in the form of the input from one electrode minus the input from a second electrode. Montages are either bipolar or referential. In order to minimize noise, a separate reference is often chosen for each side of the head. Bipolar and referential montages contain the same basic information that is transformable into either format by simply subtraction, as long as all the electrodes, including reference, are included in both montages and linked to one common reference. Every channel has an individual multi-stage AC coupled very sensitive amplifier with differential input and adjustable gain in a wide range. Its frequency response can be selected by single-stage passive filters. The preamplifier used in electroencephalographs must have high gain and low noise characteristics because the EEG potentials are small in amplitude. In addition, the amplifier must have very high common mode rejection ratio. The artifacts present in the EEG signal are generally removed using low-pass filters. EEG amplifiers are selected for minimum noise level, which is expressed in terms of an equivalent input voltage. The writing part of an EEG machine is usually of the ink type direct writing recorder. The best types of pen motors used in EEG machines have a frequency response of about 50 Hz. An accurate and stable paper drive mechanism is necessary, and it is normal practice to have several paper speeds available for selection. An EEG is recorded simultaneously from an array of many electrodes. The record can be made from bipolar or monopolar leads. The electrodes are connected to separate amplifiers and writing systems. Microprocessors are now employed in most of the commercially available EEG machines. These machines permit customer programmable montage selection. Evoked potentials. If an external stimulus is applied to a sensory area of brain, it responds by producing an electrical potential known as the evoked potential. The most frequently used evoked potentials for clinical testing include brainstem auditory evoked response, visual evoked response, and somatosensory evoked potential. Evoked potential is recorded at the surface of the brain and is the integrated response of the action of many cells. The amplitude of the evoked potential is of the order of 10 microvolts. The evoked potentials are generally superimposed with electroencephalograms. Therefore, it is necessary to remove the EEG by an averaging technique while making the evoked potential measurements. Since the background EEG and other unwanted signals often appear irregular, they can be reduced by averaging across multiple trials. Analysis of EEG. Assessment of the frequency and amplitude of the EEG is crucial for rapid and accurate interpretation. The different methods are frequency analysis, amplitude analysis, compressed spectral array, and dot density modulated spectral array. Frequency analysis takes the raw EEG waves, mathematically analyzes them, and breaks them into their component frequencies. The most popular method of doing this is called the fast Fourier transform or FFT. Fast Fourier transformation of the digitized EEG waveform is a mathematical transformation of a complex waveform having varying frequency and amplitude content into simpler, more uniform waveforms such as different sine waves of varying amplitudes. In this method, the EEG signal is converted into a simplified waveform called a spectrum. It is then separated into frequency bands at intervals of 0.5 Hz over a range of 1 to 32 Hz. The redistribution of electrical activity in the brain among certain frequency bands or the predominance of one band over the others correlates with specific physiologic and pathologic conditions. The spectral analysis transforms the analog EEG signal recorded on the time axis into a signal displayed on the frequency axis. The amplitude changes result in changes in the power of the resulting frequency spectrum. As the amplitude increases, so does the power. The most common number reflecting EEG amplitude is the total power of the EEG spectrum. In the compressed spectral array, a series of computer smooth spectral arrays are stacked vertically, usually at 2-second intervals, with the most recent EEG event at the bottom and the oldest at the top. Peaks appear at frequencies which contain more power or make larger contributions to the total power spectrum. With this method, it is easy to pick up changes in frequency and amplitude of each sample over a longer period of time, as it compresses a large amount of data into a compact, easy to read train. Dot density modulated spectral array is another method for displaying the power spectra. This format displays a power spectrum as a line of variable intensities or densities with successive epochs, again stacked vertically as in the CSA plots. Areas of greater density represent frequencies which make the greatest contribution to the EEG power spectrum. An advantage of the DSA format is that no data is hidden by the peaks as in the CSA display. Thank you.